Carbon Journal Club, I presented a paper on measuring the binding and cleavage kinetics of Argonaut proteins, and found that different AGO proteins modulate their RNA-RNA interactions differently, which led to different specificities of these proteins. And Dan noted at the time when I presented this paper that the implications were not that distinct from his 1990 paper on ribosome kinetics, more is always better. And they asked why these people hadn't cited him. And so this led, if you're familiar with this paper, the central hypothesis is that if you increase the affinity of, in this case, a ribozyme for its target, binding becomes the rate-limiting step, and so the enzyme begins to catalyze incorrect substrates at the same rate as correct substrates because it isn't able to dissociate from the incorrect substrate before it's cleaved. And so this led me and a postdoc in the lab, Inga, to scour the literature regarding RNA-guided nucleases such as AGO and Cas9, and Rick did a really good job of introducing this, where specificity is an incredibly important feature that groups all over the country and the world are trying to characterize and engineer these proteins to be specific. So we realized that researchers in both of these respective fields kept reobserving this counterintuitive fact. That is, they make their enzymes less stable in binding, they actually get more specific enzymes. And we decided that since researchers hadn't described a physical model to this process, and they certainly weren't citing Dan enough, that we should alert them to this fact and reintroduce this concept in a perspective piece, and maybe it will get cited more in Dan's original paper. And it's sort of the idea was that the strategies that the CRISPR and AGO field to basically come on was like by decreasing the length of the guides or introducing mismatches. They were decreasing or increasing the off rates or decreasing the KCAT. And so they were changing the regime of the enzyme from being in the sticky, the Briggs Haldane regime to a Cas9 regime. And so why I say this changed the direction of my career is because in this exploration, I read a lot of papers from Dave Bartel's lab, and now I'm doing my postdoc on Argonaut binding kinetics, which I never thought would actually happen. And it's also had me explore a couple job interviews in the Boston area with companies that work on RNA-guided nucleases, including CRISPR. So the last project I initiated in the Herschlag lab was a bit of a Hail Mary on my way out, which is where I think I learned how powerful quantitative energetic measurements could be in learning about the structures and conformational preferences of biological macromolecules. And so this project was in close collaboration with Sarah Denny from Will Greenleaf's lab and Joe Yesselman from Rudy Doss's lab, where we teamed up to study the conformational dynamics of RNA elements. And most of the work I'm showing you here was initiated by the three of us, but was certainly finished long after I left Stanford by Sarah and Joe. So historically, Dan had been trying to get me for, I think, probably 3 years at this point, to do a high-throughput RNA folding project, because I think he was tired of me bringing in data one molecule at a time. And so decided that he needed all the molecules at the same time. And I think in some way Dan has the perfect background for this type of project because he's really good at thinking about individual molecules deeply, but also able to conceptualize large-scale data sets, probably from his time working with Pat in the microarray days. And so to study the conformational dynamics of RNA elements, that includes helices, junctions, and tertiary contacts, what we did is we placed them in the context of this RNA-RNA heterodimer, tecto-RNA, where we put our RNA element in one of the binding partners, and then we measured binding to this new tecto-RNA variant. And in this way, we were coupling the conformational preferences of this introduced RNA element to binding affinity. And so, in collaboration with Will Greenleaf's lab, we were able to use an Illumina sequencing chip where we could express thousands of these tecto-RNA variants and measure their affinities at a single time by introducing a fluorescently labeled flow piece across the chip. And what was cool about this is we introduced these RNA elements in a bunch of different scaffolds. And so we could compare the affinities across these different scaffolds to compare the conformational preferences of these different RNA elements. This is a little convoluted, but I'll give you an example. So here in this example, we took all possible RNA mismatches and we put them in a set of scaffolds. These 9 scaffolds shown here. And then we measured the affinity to these scaffolds using a common flow piece, or the common other side of the detector RNA heterodymer. And so what we got were a bunch of these binding profiles across these different scaffolds, which we called thermodynamic fingerprints. And what Sarah did was clustered these thermodynamic fingerprints using K-means clustering. We came up with 7 main clusters. And so what we found is I'm showing the average of those 7 clusters, those thermodynamic fingerprints, across a bunch of different scaffolds and also different flow pieces. And we found this lovely relationship with just RNA mismatches that these clusters stratified based on purine permitting content of the mismatch. And so you can see in the first 2 conformational classes, they're highly enriched for GG and AA mismatches. And then the last set of clusters are highly enriched for UC and CU mismatches, with the middle clusters including the purine permitting mismatches, which are not too different than the Watson Crick elements. And so what I'm showing you is the 3D conformational behavior of these mismatches. This has nothing to do with their secondary structure energetics, which I thought was pretty cool. So what we did is we extended this analysis to doing lots of different RNA junctions, helices, and we also did a bunch of elements that have known crystal structures. And what Sarah did is she grouped these different elements by their thermodynamic fingerprints. And she also grouped them in context with elements that had crystal structure confirmation. So those are the elements that have little black lines here. And so what Sarah hypothesized was that these groups of crystal structure conformations, these ensembles, might explain the conformational behavior of all of these grouped elements, not just the ones that have the crystal structure conformations. So to test this hypothesis, Joe and Sarah used these ensembles in a Monte Carlo simulation for tecto-RNA binding, where they put different conformations of these elements based on their weight in the ensemble and simulated binding, where binding was scored as the distance between this helix and this tertiary contact here in the simulation. And what they found was that this ensemble of states was using this ensemble model for a single RNA element was relatively predictive of the measured affinities in our measured affinities of tecto-RNA. And this did much better than just using a single lowest energy state of the ensemble. And so this project was published just a couple months ago, which was a full 3 years after I graduated from Stanford. So this had the unintended consequence of I still had the privilege of Dan being in my life into my postdoc. I've often throughout this time gone to him with questions about life and about science, and he's definitely eased that transition for me tremendously. And I'm sure most trainees here can relate to the fact that Dan takes care of us far after we leave the lab. And he always has a bit of advice, whether it's solicited or unsolicited. Now I'll switch over to talking briefly about my current postdoc project in Dave Bartel's lab. And what I hope you'll see is it's a bit of a synthesis of my Herschlag lab experience. So this work is relatively new, and I've not presented it before, so bear with me. So in Bartel, we study in general post-transcriptional gene regulation with an emphasis on translation and RNA stability and the effect of microRNAs that have on these 2 processes. So microRNAs are small endogenously transcribed RNAs that get processed and integrated get loaded into this AGO family protein, and then bind to targets in mRNA 3' UTRs, leading to the recruitment of factors that repress translation or lead to the decay of the mRNA. And so binding occurs through these RNA-RNA interactions in the context of this AGO family protein, which is in this crystal structure here, where the microRNA is in red and the backbone is in green. And so what I'm going to talk about is how Argonaut modulates these RNA-RNA interactions by interpreting hyperbit binding affinity measurements. So shown here is the binding reaction between the AGO microRNA complex and the target. And we know from a lot of experiments, in vivo reporter assays, computational experiments, that what is important for this pairing is pairing to the first 8 nucleotides of the 5' end of the microRNA called the seed region. And different varying amounts of complementarity are important for repression. Jeez, really? Okay. Yeah, okay. So what
I thought you were talking about the data.
Yeah.
I thought you were quoting Dan.
dysfunctional or even increases affinity to these sites. So we employ this technique called AGO, or RNA binding seq, where we incubate our protein with a random library of RNA. Using filter binding, we pull down the RNAs that have argonaute bound, and we submit them for high-throughput sequencing, and we compare their enrichment to an input library, which I'll just skip. And we can do this over many concentrations of AGO, and what we get are these enrichment profiles that have this characteristic rise and fall, which is, I can talk about why they do this later if you're curious about this. And what we did, which is different than a lot of other labs that do RBNS, is we were able to devise a computational model for the binding of all the population of RNA molecules in this reaction. And this allowed us to get quantitative measurements of KDs that explain these enrichment profiles. And so what I did was I enumerated all the things that I thought would matter for these sites that involve 3' pairing affinity. And the two things that I want to point out for the rest of this talk is that the length of these 3' sites we thought to matter, so the varying amounts of complementary 3' end, as well as the number of nucleotides that were looped out between the seed and 3' pairing regions. So I call these things loops. And so we can enumerate all these sites and get a measurement of 20,000 different KDs in a single RBNS experiment, which takes about a week to do. And so what I'm showing you here is the affinity of a site that contains 5 nucleotides of pairing across different loop lengths. And in black, this is a site that contains a mismatch in the seed region. And so this is the affinity of just this site alone with no 3' pairing. In green is the affinity of a site that includes 6 nucleotides of pairing with the seed, and in red is the 8-mer affinity. And so what we find is we increase the length of pairing, we get an increase in affinity. And this sort of dies off after 7 or 8 nucleotides of 3' pairing. And so there's a lot of information in this graph, but we can come to a couple of major conclusions. One is that we get about a 100-fold effect in affinity from this seed alone site compared to one that has 3' pairing affinity. And that after a certain length, there's no additional affinity imparted for increasing number of base pairs, which suggests that the protein is inhibiting some amount of pairing after a certain point, which is consistent with the crystal structure of AGO, where the 3' end of the microarray is tucked away to the plasmin. And it also, this number of 100-fold per 7 nucleotides of pairing is much smaller than you would expect for a duplex in solution. And so what this suggests is that argonaute is inhibiting access to the 3' end of the microRNA, which is also consistent with what people see in crystal structures. Also, that argonaute can accommodate particularly large loops. So these are loops of 10 to 12 nucleotides in length. And data I'm not going to show you today, we can suggest that this is a little bit like a freely floating single-stranded RNA molecule in solution. And it's not making particular interactions with the protein. And with that, I'll finish here. Yeah, this is stuff that I'm doing, and I'm excited that I get to work with Dan on some future work studying RNA forming models in vivo. And I'll acknowledge both the labs, my collaborators, and these people, and my company. Questions? Thank you for sticking to the crowd. Are there any questions? Yes?
So, there tends to be a disconnect between, so you pick up a lot of these sort of 3' pairing sites, if you immunopurify the argonaute, but it seems to be disconnected from functional effects.
Yeah. So some of those experiments involve ligation experiments where they're looking at, you're talking about these chimeric ligation.
Oh, crapseed or whatever it's
Yeah. Crapseed. Where they're looking chimerically between the target and the seed. And I think what happens in those experiments is actually 3' end in a conformation that allows for ligation to happen. And so you get a higher proportion of those types of sites in those experiments. And CLIP ends up being not very predictive of repression in a general way.
Okay. And then the other thing, I think if you have a lot of pairing in the 3' end, didn't Bolzamor or Ian McCrae show that it can eject the argonaute?
Yeah.
So
Yeah.
do you see evidence Or, well, I don't know how you would measure that.
I
If you
can't
see it,
measure
I
that,
guess you-
but we find that some microRNAs have significant 3' pairing, and that those microRNAs may be more prone to something called target-mediated degradation, where if they have extensive 3' pairing, they actually dislodge the microRNA. And so we have a model that some of these microRNAs might be prone to that.
I just have, what are typical off-rate ranges for the RNA?
1 times 10 to the -6 per second, 1 times 10 to the -8 per second are things that, so like, we're like picomole, yeah, they're pretty tiny. I incubate these reactions for 8 hours, and I do long time points just to make sure that
Okay, so if there are any more questions, you can approach me that I do. Now, our next speaker is Inga. Inga is a current professor in dance group.